morning, everybody. In today's talk, we shall see how the inanimate, inorganic world that existed before life began converted into an organic world when autotrophs came into existence. Let us begin then. Carbon is a unique element which is seen in all organic compounds. Now, carbon is unique because it can form both inorganic as well as organic compounds. In inorganic compounds, it can exist as carbon dioxide or bicarbonates, or in organic compounds, carbon forms stable covalent bonds with either hydrogen or with another carbon. So these two types of bonds, that is CH or CC bonds, are unique to all organic compounds. Besides these bonds, organic compounds may also incorporate other elements like nitrogen or phosphorus in them. Today we know over 9 million organic compounds, and carbon dioxide is thought to be the source for most of them. So how do living organisms get carbon? As seen earlier, autotrophs are able to convert carbon dioxide into organic compounds, and heterotrophs, they depend directly or indirectly on these autotrophs for acquiring carbon. The process of acquiring organic carbon, using it for synthesis of complex compounds like carbohydrates, proteins, nucleic acids, lipids, and further breaking down these organic compounds to release energy, is called metabolism. All living organisms need metabolism to maintain their living state. So in this talk, we shall study the different types of autotrophs, namely chemoautotrophs and photoautotrophs. We shall study how energy is obtained for carbon dioxide fixation, how light energy is captured by photoautotrophs, how carbon dioxide is fixed, and how the fixed carbon is used for metabolism. So firstly, let us study something about autotrophs. Autotrophy is the capacity of an organism to derive all cell carbon from carbon dioxide. All autotrophs require a source of energy and a source of electron donors. And of course, the source of carbon, which in autotrophs is always carbon dioxide. Now, there are two types of autotrophs known. Firstly, the chemoautotrophs use inorganic chemical reactions as a source of energy, and the electron donors could either be hydrogen or reduced inorganic compounds. Whereas photoautotrophs, they use light as the source of energy. Now, in anoxygenic photoautotrophs, reduced inorganic compounds are used as electron donors, whereas in oxygenic photoautotrophs, water is used as an electron donor. The process carried out by photoautotrophs is called as photosynthesis. Now, carbon dioxide fixation, as we know today, is carried out by protein catalysts, which are enzymes. But these proteins are also organic compounds. So how could carbon dioxide fixation take place before proteins evolved? It is thought that minerals like pyrites could have catalyzed carbon dioxide fixation before the proteins existed. So let us study the autotrophs in more detail. Life is thought to have originated in deep oceans, in ecosystems called as the hydrothermal vents. Now these vents represent regions where the molten lava from the core of the earth spills into the oceans and solidifies. Now in these vents, there is no light energy or no oxygen available. Yet organisms do grow in such an environment. And these organisms are the chemoautotrophs. Chemoautotrophs are able to obtain energy by two types of reactions. One is the reduction reactions, wherein hydrogen is used as a source of electrons for reducing carbon dioxide or sulfur. Now, when carbon dioxide is reduced using hydrogen, it forms methane. And when sulfur is reduced by hydrogen, it forms hydrogen sulfide. Now, methane formation is carried out by organisms called as methanogens, and they belong to the kingdom Archaea, whereas hydrogen sulfide formation is carried out by both bacteria as well as Archaea. Now, some of the chemoautotrophs also use oxidation reactions. In this case, they use hydrogen sulfide, which is abundant in this environment, as a source of electrons, and oxidized ion compounds, namely the ferric compounds, as acceptors. So hydrogen sulfide reduces ferric to ferrous sulfide and forms protons. So these type of oxidation reactions as well as the reduction reactions release energy, and this energy is used for carbon dioxide fixation, about which we shall study slightly later. But these anaerobic redox reactions are thought to represent the most primitive type of autotrophy. Photoautotrophy is when organisms use light energy to fix carbon dioxide. The process is called as photosynthesis. Now, photosynthesis could have originated in shallow lakes because light could penetrate through the water of such lakes. There are two types of photosynthetic organisms. Some organisms carry out anoxygenic photosynthesis, where light is used to oxidize inorganic compounds like hydrogen sulfide to sulfur. The energy released in this reaction is used to fix carbon dioxide. Now, this anoxygenic photosynthesis is quite similar to what happens in the chemoautotrophs, where hydrogen sulfide is also oxidized to sulfur, but in chemoautotrophs, ferric ions are used as the electron acceptors, whereas over here, the hydrogen sulfide is directly oxidized to sulfur by using light energy. Now, both these chemoautotrophs and anoxygenic photosynthesis is thought to have evolved about 3.5 billion years ago, before Earth's atmosphere was oxygenated. What is the evidence for this? They have detected recently microfossils of microbes, and the rocks in which they were found dated back to 3.5 billion years ago. So we can say that these processes probably existed 3.5 billion years ago. The microbes present in these microfossils were methanogens as well as the chemoautotrophs, which we have seen in these slides. Chemoautotrophs also play an important role in biogeochemical cycling of elements, but we shall not go into that aspect here. The next type of photosynthesis is called as oxygenic photosynthesis, where water is used as a source of electrons. So light energy converts water into oxygen. So this type of a unique reaction led to the oxygenation of the atmosphere, and this was present in cyanobacteria present in the shallow lakes, which used light energy for oxidizing water to form oxygen. Now, this accumulation of free oxygen in the Earth's atmosphere is thought to have taken place 2.5 billion years ago. And the evidence for this is that many of the iron compounds which were soluble in the lake waters were seen to precipitate due to oxidation by the oxygen liberated by the cyanobacteria. So this process was known as the rusting of the Earth's oceans or lakes. Now, what is the fossil evidence to prove that this process took place 2.5 billion years ago? They have found fossils called as stromatolites, which consists of cyanobacterial mats entrapped in layers of minerals. And the dating of this rock is 2.5 billion years ago. So this oxygenic photosynthesis is very important because it has enabled present day life on Earth. We shall come back to this slightly later. In the earlier slides, I have told you that energy from chemical reactions, namely the redox reactions, or from light, is used for fixing carbon dioxide. So how is this energy harvested? It is harvested by the formation of a proton gradient which forms across the membrane. The potential energy present in this proton gradient, that a proton gradient would be that there are more protons on one side of the membrane and less protons on the other side of the membrane. So it forms a gradient. So there is potential energy present in this gradient, and when this potential energy is converted to chemical energy, ATP is synthesized. So let us see something about this process, how this process occurs by which a proton gradient arises. Let us take an example which we have seen in the earlier slides, namely of an organism which is able to convert hydrogen sulfide to sulfur by passing the electrons to ferric compounds. So ferric is reduced to ferrous, and in the process protons are liberated. Now these protons accumulate on the outer side of the membrane because organisms carry out this process of oxidation of H2S outside the cells. Now the electrons from this reaction are accepted by ferric compounds present within the membrane, and they are reduced to form ferrous. The ferrous in turn, on the cytoplasmic side of the membrane, they use electron acceptors like ferrous, like oxygen, where the reduced ferrous passes the electrons to oxygen, and protons are also used in the process to form ferric and water. The ferric then again is used for reduction on the outer side of the membrane. So you see that as a result of this, more protons accumulate outside, and protons on the cytoplasmic side are used up for forming water. So this leads to the proton gradient. Now earlier, before membranes existed, the pyrites themselves were thought to be redox carriers, and over here they carried out the conversion as shown over here of ferrous to ferric. After this, the protein-based redox carriers appeared and took over this function. Now even today these proteins use ferric-ferrous conversions for transferring energy, transferring protons from one side to the other. Now the proton gradient is created, but how is it used for synthesizing ATP is what we will see here. ATP synthase is a wonderful molecule because it is able to convert the potential energy created by the proton gradient to mechanical energy. So it is like a machine. It is called as a molecular machine. So you see in this equation that ADP and inorganic phosphate is converted to ATP and water, making use of the energy from the proton gradient such that the proton gradient is neutralized. So initially there will be more protons outside and less inside, and when ATP is formed, the protons on either side are the same. Now this ATP synthase consists of two subunits, the F0 and F1 subunit. The protons, when they pass through the F0 subunit, they bring about rotation of the ATP synthase. And this rotation is important because it changes the shape of the enzyme such that ADP and phosphorus can enter and ATP can form and exit from the active site. Now ATP is also synthesized by ATP synthase in mitochondria during the process of respiration. In this case, energy conversion from sugar to carbon dioxide is used for forming the proton gradient. We shall not go into the details of respiration here. Now ATP, it is called as the energy currency of a cell because like money, it can be exchanged for carrying out different types of reactions in an organism. So we use the money currency to buy various types of things, we use ATP to carry out different types of reactions. After having seen how the energy from redox reactions or from light is used for fixation of CO2, let us now come to oxygenic photosynthesis. And here I will begin the story of oxygenic photosynthesis. This process was first shown by cyanobacteria, later by algae and the entire plant kingdom. So how was this process discovered? We will go briefly into the history of the discovery of photosynthesis over here. In 1774, a scientist called Priestley discovered that plants could restore air that was injured by a lit candle or mice kept in a closed jar. So what did he do? He took a jar, a bell jar, in which he placed a lit candle and a mouse. After some days, it was seen that the candle was extinguished and the mouse was dead. So that means he said that the air seemed to have been injured due to the lit candle and the mouse and led to the death of the mouse and extinguishing of the candle. However, Priestley found that if he kept a plant in the same jar, then the lit candle and the mouse remained working for a longer time. So the plant appeared to restore the air which was injured by the candle or the mouse in this process. A few years later, another scientist called Ingenhousz in 1779 discovered that in the presence of light, plants give off bubbles from their green parts. So as seen in this experiment carried out here, branches of hydrilla, which is an aquatic plant, were taken in a beaker of water and a funnel was inverted over it. Now on the funnel was placed a test tube. When this whole apparatus was exposed to light, it was seen that air bubbles were evolved and they accumulated in the test tube. Now Ingenhousz called this as combustible air because he did not know what the composition of that air was. But he knew that it could support combustion. At the similar time, Lavoisier discovered oxygen. This was in 1777, and later people found that the gas which is liberated in the experiment of Ingenhousz was indeed oxygen gas. Further, in 1796, a scientist called Senevier showed that plants not only released oxygen with the help of sunlight, but they also absorbed CO2 through stomata. And finally, in 1804, a scientist called Saussure showed that plants took in carbon dioxide from the air and water from the soil and liberated oxygen. Now in this process, the plants grew, they gained weight, they gained biomass. So that means carbon dioxide and water were used to add to the biomass of the plant and oxygen was released. So where does this photosynthesis take place? Now eukaryotic photoautotrophs like plants and algae, they show organelles called chloroplasts in the cells. It was in 1837 that a scientist called Hugo von Mohl saw discrete bodies within the green plant cell, which were later called as chloroplasts. Another scientist called Schimper in 1883 saw that the chloroplasts resembled the cyanobacteria, and later it was thought that the eukaryotic cells ingested these cyanobacteria which formed the chloroplasts or the organelles, chloroplasts. Now what do these chloroplasts look like? Chloroplasts are double membrane organelles. They consist of a matrix which is called as stroma, and they have the pigments located in membranes, in an internal membrane system which is present as stacks of thylakoid membranes which form the grana. After having seen something about chloroplasts, the next question is, how do chloroplasts capture light? So light capture is known to take place by pigments which are called as bacteriochlorophylls or chlorophylls. Chlorophyll was first isolated by two scientists, Caventou and Pelletier, in 1817. Now these chlorophylls are known to absorb light in the blue and the red region. But besides chlorophylls, there are other pigments which are also present in the chloroplasts, which are called as accessory pigments. They serve as antennas because they capture light energy of different wavelengths, not only red and blue like done by which is done by chlorophylls, but even the green and the yellow wavelengths can be captured by these accessory pigments, and they transfer their energy to the chlorophyll molecules by a process called as resonance transfer. Now to the right is shown the structure of a chlorophyll molecule. This chlorophyll molecule consists of a ring called as a porphyrin ring, which consists of 4 heterocyclic nitrogen compounds which are bonded in the center by magnesium. Now this structure is very similar to that of the compound called as heme, which is present in hemoglobin, except that in hemoglobin iron is found to be bound to the 4 porphyrins which are present in heme. Now chlorophyll also shows a long tail of hydrocarbons which is called as the phytol tail, and this phytol tail holds the chlorophyll pigments within the membrane. So we know that red and blue wavelengths are captured by chlorophyll, but how do we know that they are used for photosynthesis? In 1881, a scientist called Engelmann, he devised a very interesting experiment. He placed a green filamentous alga under a microscope and illuminated it with a light spectrum which he obtained using a prism. So as seen over here, the filamentous alga was placed on a slide and the spectrum of light was, and the slide was exposed to a spectrum of light. Now on the slide were also some aerobic bacteria which were present in the medium around the green alga. Now when he observed their distribution after some time, he could see that the aerobic bacteria, they aggregated in regions which were illuminated by blue light and by red light. So that means the photosynthetic alga liberated oxygen, maximum oxygen in the blue and red light, which attracted the aerobic bacteria to grow around these regions. So a very simple and nice experiment was carried out by Engelmann to show that red and blue wavelengths are used for photosynthesis. The next question is that what happens when light energy is captured by chlorophyll? As seen earlier, there are antenna pigments. These pigments are present in a complex called as the light harvesting complex, and when they absorb light, they transfer all the energy, the light energy, to a special chlorophyll present in what is known as the photosynthetic reaction center. Now this chlorophyll is special because when it receives all the light energy from these accessory pigments, it ejects electrons from the chlorophyll molecules. Further, the high energy electrons which are ejected from chlorophyll are transported to electron acceptors. So there is electron transport taking place. This scheme was worked out by two scientists, Hill and Bendall, in 1960. So a long gap before the mechanisms of photosynthesis were discovered. Now once the electrons are accepted from chlorophyll by electron acceptors, the electrons travel downhill. That is, downhill in the form of electropotential, and they are passed down the hill by accepting and releasing energy in a stepwise manner. So some energy is lost as the electrons move down this electron transport chain. This energy is used to generate a proton gradient and ATP. Finally, the electrons are used to reduce a compound called as NADP. So you get reduced NADP, which is called as NADPH. We won't go into the details of that. But remember that two electrons have been ejected from chlorophyll in the presence of light. So how will this chlorophyll continue its function of light capture and excitation? It is here that water comes into the picture. Now oxidation of water is used to provide the electrons to this chlorophyll so that it fills up the hole which is left in chlorophyll. Now due to oxidation of water, oxygen is liberated as a byproduct. So the three products of the light reactions can be considered to be ATP, which is formed from a proton gradient. Then you have NADPH and you have oxygen. Now what is this ATP generated in light used for? This ATP is used for fixing carbon dioxide. This was shown by a scientist called Kelvin in 1957. Now he used a radioisotope of carbon, namely C14, and this radioisotope emits beta particles which can be seen when exposed to an X-ray film. So using this radioisotope of carbon, he devised an experiment where he took an algal culture in a vessel and placed this in light. Then he injected the radioactive carbon in the form of bicarbonate, which is a source of carbon dioxide for the algae, and very quickly he opened the valve at the base of the vessel to take a bit of the algal suspension into hot methanol. Now this suspension, which was harvested in hot methanol, was then chromatographed on paper, and this paper was exposed to an X-ray film. So within 5 seconds he saw that one of the spots on this chromatograph showed an exposure, okay? And this spot was due to the formation of a compound called as 3-phosphoglyceric acid. So within 5 seconds C14O2 was consumed and it formed the compound 3-phosphoglyceric acid. Later on, the radioactive carbon was seen to be present in a large number of compounds like amino acids, organic acids, and etc. So in 30 seconds, the carbon which was taken in by the algal cell was used to synthesize first phosphoglyceric acid, and then this phosphoglyceric acid probably was used for the formation of various other compounds. Now this technique is called as autoradiography. So using this technique, Kelvin took aliquots or small parts of the algal suspension at various times, and he found gradually what this PGA or phosphoglyceric acid which is formed, it is used for. So he found that the first step in carbon dioxide fixation is that carbon dioxide binds to a compound, a 5-carbon sugar called as ribulose bisphosphate. And the enzyme which binds carbon dioxide to this 5-carbon sugar is an enzyme called as ribulose bisphosphate carboxylase. It is the key enzyme in this carbon dioxide fixation pathway. Now once this 6-carbon compound is formed, it immediately splits into 2 molecules of 3-phosphoglycerate. Now once the phosphoglyceric acid is formed, it is then converted to glyceraldehyde 3-phosphate using the energy from light reactions, namely ATP and NADPH are used at this step to form the aldehyde from the acid. That is a reduction reaction. Now it is this glyceraldehyde 3-phosphate which then enters the cycle for synthesis of sugars. However, there is a requirement for regenerating the ribulose bisphosphate so that the cycle can begin again. And so some of the glyceraldehyde 3-phosphate is used to form ribulose bisphosphate again so that it is ready once again to bind to carbon dioxide and the cycle can continue. The sugar which is formed in this cycle is used to synthesize starch, which is a storage product, or the sugar is transported to other parts of the plant where photosynthesis does not occur, like the roots or the flowers or the fruits, etc. Now once this carbon dioxide has been fixed and converted to 3-PGA and then to sugars, the next steps are common to all living organisms. That is, autotrophs and heterotrophs use this sugar to carry out metabolism. So there are 2 types of reactions which characterize metabolism. One is called as anabolism, where the sugars are used for synthesis of complex molecules like carbohydrates, proteins, lipids, nucleic acids. Now this synthesis, this biosynthesis of complex molecules requires an input of energy in the form of ATP. Now, from where does this ATP come? There are some reactions going on called as catabolic reactions, wherein the compounds synthesized during anabolism are broken down into simpler compounds, and this process is called as respiration. So during respiration, the sugars or the complex compounds formed from the sugars are broken down and carbon dioxide is liberated. In this process, ATP is synthesized, which is used both for the anabolic reactions, because anabolic reactions require input of ATP, and also ATP is used to carry out other processes like movement or excretion or transport. So this anabolic and catabolic reactions are known to take place in all living organisms. To summarize then, we have seen that organisms show 2 lifestyles, namely autotrophy and heterotrophy. We have studied 3 types of autotrophic processes, namely chemoautotrophy, anoxygenic photosynthesis, and oxygenic photosynthesis. We have seen that the energy through redox reactions or from light is used to form a proton gradient across membranes, and this proton gradient, the potential energy of this proton gradient is harvested for the synthesis of ATP. The harvest of light energy by the photoautotrophs is used for fixing CO2 to form organic carbon. We have seen how the Calvin cycle does this. We have also seen how the organic carbon, which is used by the autotrophs and heterotrophs for metabolism. So from all these slides, you will appreciate that autotrophs are at the base of the organic world. They either provide sugars to the heterotrophs and enable all life which exists on earth today. Now, could autotrophs exist on Mars? This is where I want to end the lecture. Now, recently, several rovers have been sent to Mars to look for signs of life on that planet. The rovers have already detected the presence of methane, hydrogen sulfide, and water on Mars. Now they are looking for the possibility of autotrophic microorganisms that could possibly have lived or are living on Mars. Why? Because we have seen that methane, hydrogen sulfide, and water are either substrates or products of the chemoautotrophs or the photosynthetic organisms. So in future you may hear that Mars is inhabited with one of the types of organisms that lived on the early Earth. I'll end here. Thank you very much.